All right. I believe we're on part 3 now. Let's move on. It might be part 4. I kind of lost track. Okay. Hot air ovens. So we already mentioned that if we're going to try to sterilize in a hot air oven, we need to have a much longer amount of time. So this is just giving us an example. If we're putting things in this hot air oven at 150 to 180 degrees Celsius, then it's going to take 2 to 4 hours. But essentially, it's going to be dependent on that temperature. So we do have to have special ovens that get that hot. And what we see is we are able to decontaminate and sterilize that equipment. Now, one thing to mention with both the dry heat technique and the autoclave or the moist heat technique is we don't want to destroy any the properties of whatever we're trying to sterilize. So there are certain substances that would not do so well. So you can't put plastic, for example, in an autoclave. It's going to melt. So if you're trying to sterilize plastic, you might try, the dry heat methods as long as that plastic can resist, up to 180 degrees Celsius. So these methods really are dependent on what it is you're trying to decontaminate or disinfect. All right, effects of the cold. So opposite of hot is cold. And essentially what we're going to see, this is showing us percent maximum microbial activity at these various temperatures. So we can see that our psychrophiles are those that like it really cold and those that like it warmer. Thermophiles, essentially, you're not going to have a whole lot of organisms that are essentially growing at those temperatures, but there are some that do. And as I mentioned before, refrigeration is bacteriostatic, so we're just trying to keep the level of microorganisms down so our food doesn't spoil so quickly and it doesn't harm us. Freezing can be either bacteriostatic or, depends on the bacteria, could be bactericidal and kill those bacteria, just depends on which microorganisms we're talking about. Now, sushi, when you go in and you think you're eating raw fish, you are eating raw fish, but in the US, the FDA requires that raw fish for sushi be frozen negative 20 degrees Celsius for 7 days or negative 37 for 15 hours to ensure that that fish is free of microorganisms. Turns out that that particular method does not alter the fish at all. So you're able to have your raw fish sushi without it actually being compromised as far as taste, because essentially that fish is still preserved and its texture and flavor is maintained. Okay, desiccation is an interesting thing. So I have look at my face somewhere. I have a food dehydrator at home. And so when we're thinking about food dehydration, maybe it's like I first did put strawberries in there. I pitted cherries and put cherry halves in there. Oh my gosh, those are so good. Apples, pears. You can essentially desiccate all kinds of food, right? Desiccation is doing an interesting thing because it's removing the water. And when you remove the water, you're increasing the solutes. So you're increasing the other chemicals, if you will, inside the cell. So once we do that, we create a situation of high osmotic pressure. Okay, high osmotic pressure is essentially going to be microbistatic or bacteriostatic, simply because most organisms cannot live in a high solute situation. So that's what's going on there. And freeze drying. So you might have had camp meals. I know we used to do that until we realized you only really need that for backpacking. But when we have these freeze-dried foods, it's a flash frozen food. And essentially, again, that's going to lead to a bacteriostatic situation because we are drastically reducing the ability for that bacteria to grow, right? So freeze drying is another way to essentially increase that osmotic pressure and preserve the food so it still tastes good. And then it turns out that we can also order bacterial cultures here. to the campus when we're needing them for various labs like genetics or microbiology, cell biology. And so it is a way to actually preserve bacterial cultures. So it may not always be negative towards bacteria. Okay, radiation methods. Let me put my face over here. Radiation methods. So we take a look here at the visible light spectrum, which is just this tiny little part of full radiation. So you also have equivalent to that then is as we decrease the wavelength, you increase the energy. So as you increase the wavelength, you decrease the energy, which is why radio waves and microwaves are not necessarily harmful to us. But we know when we get down to this increasing energy range, and we're talking about ionizing radiation, that it certainly can be used as a physical control method simply because we are bombarding microorganisms with these rays. So I'm thinking about this ionizing radiation. So we're killing the microorganisms. Okay, so X-rays, gamma rays, electron beams, essentially what they're going to do is they're going to damage the DNA in our microorganisms, penetrating them, killing them, and so that's how we're using that ionizing radiation to essentially sterilize, or at least, at the very least, we are able to disinfect or decontaminate. Okay, let's take a look at non-ionizing radiation. So this is UV light. So thinking about UV light. So when we go outside, this time of year when it starts getting warmer, we get a little sun because we're just outside gardening or taking a walk with the dog. So the interesting thing about these longer wavelengths of radiation is that they are non-penetrating. So it just damages essentially, like for us, the surface layer. So just for example, the epithelial cells of our skin. So if we go to the other end of the spectrum where we have these short waves with very high energy, the ionizing radiation is able to penetrate very deeply into materials. That's why, like X-rays, we want to use them when we need them. We're going to use protective mercury. Is it mercury? I don't know. Anyway, right now I can't think. But essentially, you have those big, heavy metal drapes that you put on parts of your body so that we're not causing any damages to DNA, for example, in the breast or in your testes and ovaries. Microwaves up here, not harmful, also not going to kill microorganisms. All right, wanted to tell you a little bit about gamma irradiated meat. So gamma irradiated meat is a way to essentially ensure that our meat is going to last longer. So to give you an example, irradiated beef can last 42 days in the refrigerator compared to regular beef, which is about 5 days. It's not dangerous. There's not any residual gamma rays in that food. Essentially, it's used to irradiate the meat when you're not around, and it's as soon as that the gamma rays are turned off, then there's no residual residual radiation in that beef. So it's a really great way to essentially be able to buy a reasonable amount of beef at a good price and then be able to keep it in your fridge for longer so that it's not spoiling. We also can do UV irradiation for drinking water. So once we looked at a house, I think it was just on the west side of Watkins Glen, and I guess the water was known to be contaminated with microorganisms, so they had a very elaborate water filtration system in the basement, which included reverse osmosis, and then finally it was UV irradiated. So it would kill any microorganisms that would be you would potentially be exposed to. Just looking at my notes for a second. Okay. So we should see some apple cider there as well. All right. This is kind of interesting, the Xenix robot. So think about those infectious diseases that are in rooms in a hospital because the patient has them, right? So it could be, all kinds of things, yellow fever, it could be tuberculosis, it could be COVID, it could be, all kinds of things. So one thing that they do is they send in these robots that essentially is going to move around the room, exposing the surfaces to UV radiation so that it's killing any residual amounts of the microorganism or the life cycle of the microorganism so that then that room is now completely clean. Now, a benefit of that is that we're not sending a human in there to do that and potentially be exposed. So kind of a neat way of using our technology to the best of our interests here and ensuring that hospital staff as well as future patients that might be in that room are not harmed by an infection. All right, I believe this is our last thing, filtration. So your book put it into mechanical methods, but it's essentially a physical type. But filtration using membrane filters, essentially think about HEPA filter, so your vacuum cleaner. When we vacuum, essentially there's a series of filters that's trapping pollen, other kinds of allergens like pet dander, even like the larger holes would trap hair, right? So you have a series of membranes with large to small openings that essentially is going to filter out various particulate matter. And then essentially then we're going to get down to a very thin membrane with small pore sizes, and that's going to be filtering out those really small things. Like I'm thinking pollen, dust, dander from your animals. So membrane filters are very useful and really just kind of this passive way of ensuring that we are decontaminating substances. So we use it to prepare liquids sensitive to heat, such as serum, blood products, vaccines, drugs, IV fluids, enzymes, and media. So blood, that's a very important thing. When you give blood, they are going to send it through filters to ensure that you're not that they're not giving that to a patient who then is going to contract something from your blood. We know that membrane filters are alternative methods for decontaminating milk and beer, so don't have to alter the flavor. We can use it in water purification, so most of us on well water have a whole house water filter. You can have a series of water filters so that you can eliminate smaller and smaller particles. And so that's pretty neat. The last thing filters out particulate impurities like crystals and fibers and so on that can cause severe reactions in the body. So you might be wearing a special mask, for example, when you're working with asbestos. This shows us a HEPA filter, the pre-filter, carbon filter, and then essentially then when you get to the actual HEPA filter, it's doing the very smallest particles. Liquids, we can actually sterilize or eliminate, if you will, microorganisms from liquids by using a vacuum and filtering the fluid through a membrane. So if I didn't have time to heat up the autoclave, I could quickly do this in the lab so that we would have media for lab. All right, the very last thing I wanted to talk about was what we learned from COVID. So I wanted to apply this to what we know about filters. So a surgical mask essentially is designed to reduce droplets from the wearer. So you're wearing it, it's protecting others from you. So essentially what it's going to do is it's going to prevent your sneezing or coughing and putting droplets in the air. Now, when individuals wear an N95 respirator or mask, like I had to do when I was in both working at the ICU and a local nursing facility, is essentially what we're doing is we're blocking 95% of the airborne particles that would enter in and then expose us. So when you wear an N95 mask, it's protecting you from others. And turns out that that N95 mask is a type of HEPA filter, decreasing essentially particulate matter so that you are not being exposed to airborne infectious materials or particles. And we're thinking about the teeniest, tiniest little ones. So again, what I suggest that you do, take some time, fill out the 2 tables that are in this part of your handout. And I will be posting a key in the next day or so. And that'll help you condense this material. And again, you're going to move on to Dr. Gustin's lectures on chemical controls. See you later.